First, thanks for the introduction. And I think we made another diagnosis here is that Lee and her brother RJ have a gene for being extremely articulate and well-spoken. It's pretty remarkable in such young people. So I'm going to talk about an iPS model of Barth syndrome. So the Barth Syndrome Foundation has invested heavily in making research models, and I think everyone in this room understands why. The progress really has been pretty rapid in this scope of things. The syndrome was diagnosed in '86. The TAS gene was discovered as the cause in '96, and in the next decade, really, we established models in all of the major animal systems, and much of this work was done by the people in this room. When I started this work in 2010, there was not yet a mammalian model of Barth syndrome. Now we have the mouse model. But one of the goals was to establish a model that would recapitulate the physiology of heart muscle cells. This did not work. So I was inspired by this really transformative work by Shinya Yamanaka, who made the discovery that you could reprogram skin cells into stem cells. And of course, this was published in 2007, and in a remarkably short amount of time, only 5 years, this study led to a Nobel Prize, just showing how transformative that work was. So in the iPS paradigm, the goal is to take patient-derived fibroblasts, which have the genetic material of the patient, and turn them into stem cells. These stem cells can then be used as patient-specific disease models, and in the future, potentially could be modified and put back into the patients as autologous replacement therapy. But the replacement therapy part is a little science fiction right now, but what is not science fiction is the ability to use these cells as disease models. However, there's still many challenges when we consider using this technology as a model. So important things that need to be addressed are how to control for interline variation, how to control for inter-individual genetic variation that doesn't have to do with the gene you're interested in, and how to develop in vitro systems that will model the pathophysiology, in this case, the pathophysiology of a cardiomyopathy. So in this talk, I'll go over how we address those challenges and how we use this model to try to get some new insights into Barth syndrome. This is just an outline of the talk. So first, I'll show the cell lines that we made. We made 2 iPS-derived cell lines. One is called H, one is C. One is a frame shift mutation, the other is a point mutation. We used 2 different reprogramming methods. We did the standard quality control, including karyotype and teratoma assay. We then learned how to differentiate these cells into cardiomyocytes. This is a now very commonly used protocol to make cardiomyocytes from iPS cells. We were able to use magnetic bead purification to get an 80% pure preparation of myocytes. This is what a stem cell looks like, and then this is what the purified sheet of cardiomyocytes look like. They're stained with the marker of a cardiac protein. And if you put these cells on a piece of plastic, they'll form these beading sheets. So next, we asked if these cells could recapitulate the metabolic and contractile abnormalities of Barth syndrome. And you're all very well aware of the biochemistry in which we have too much of this immature form, the MLCL. And TAS converts the immature form into the mature form of cardiolipin and shown on a mass spec. And in Barth syndrome, we see an excess of the immature form and a reduced mature form, and the the ratio, as we heard yesterday, is diagnostic of this disease, and the Barth syndrome cells recapitulated the increased ratio. So next we looked at the metabolic abnormalities of these cardiomyocytes, and we used this assay platform called the Seahorses Biosciences Extracellular Flux Analyzer. Sounds fancy, but basically it's just an oxygen recording machine. So you plate the cells and you record their oxygen consumption. So here's how, this is the electron transport chain, and this is the basal level of oxygen consumption. If you inhibit ATP synthase, you depress the oxygen consumption. So that's the amount of oxygen that's being used to drive ATP synthesis. You then permeabilize the inner mitochondrial membrane, and you get the maximal electron transport chain activity. And then you block the electron transport chain itself, and then you block all mitochondrial oxygen consumption. So with these different parameters, you can calculate what different parts of the mitochondria are doing. So we applied this technology to regular iPS-derived cardiomyocytes, and I'll point out we did this culture in galactose, which is important because galactose does not robustly support glycolysis, so it makes the cells more dependent on their mitochondria. And compared to the control cells, the Barth cells consistently had a higher basal level of oxygen consumption and a depressed peak oxygen consumption rate. And this was a little surprising to us that the Barth cells would actually consume more oxygen at baseline. So we looked at the ATP content of these cells, again cultured in galactose, and we saw the ATP content was about half of the normal ATP content. But interestingly, if we put the cells in glucose, we could restore the ATP content because the cells were then able to use glycolysis. And we'll use this manipulation to interrogate the effect of ATP depletion on these cells. So just to summarize the results, So we had the two findings of increased basal oxygen consumption and decreased basal ATP. And so that led us to hypothesize that this ATP synthase complex was not functioning properly. And so we measured the specific activity of ATP synthase using an immunocapture kit. And we found that the specific activity of the complex was reduced. So that sort of accounts for why you might have basal increased oxygen consumption and yet have reduced ATP. And just to summarize the metabolic abnormalities, we found, of course, the impaired cardiolipin maturation, reduced whole cell ATP and galactose, inefficient ATP synthase, and reduction of the peak electron transport chain activity. So next we wanted to address this question that has really plagued the iPS modeling field of how do you address interline and inter-individual variation. And I think that this is really important to develop the right controls so that you know you're really looking at the physiology of the gene you're interested in. So we translated this question into, is the TAS mutation necessary and sufficient to give you the phenotype you're looking at? So necessary means if we replace TAS using a technology called modified RNA, then you can rescue the phenotype. And sufficient means if we take the TAS mutation and put it in a wild-type cell, can we make the wild-type cell behave like the Barth syndrome patient cells? So first we used a technology called modified RNA to put TAS back into cardiomyocytes. Here we just made a fluorescent protein called GFP with this TAS, and we showed that if we put it on myocytes, the myocytes take it up and robustly express GFP. And here we did that with TAS, and you can see that the flag tag TAS localizes to the mitochondria of an iPS-derived myocyte. And this manipulation rescues the cardiolipin deficiency of the Barth syndrome cells. So next we asked about the metabolic dysfunction of these cells. Could the TAS modified RNA rescue that phenotype? So here's the control and the mutant cells. We put the TAS-modified RNA on the cells, and we showed that we can completely correct the oxygen consumption abnormality. And we could also rescue the ATP deficit. So next, we wanted to ask the question of sufficiency. If we put this mutation into a wild-type genome, can we make that wild-type cell behave like a Barth syndrome patient cell? And we used a technique called genome editing to achieve this. And this is a technology that was pioneered by our collaborator, George Church, and is now becoming widely adopted. And I won't go into the details. I just say that we made three genetically identical cell lines, except that they have a different mutation at the TAS site. So we made one cell line that has a wild-type TAS. It was handled in the same way as these cells. Another that has the H-line TAS mutation, and another that has a frame shift mutation at the H location. And these different mutations are shown here. Over here, I'm just showing you how efficient this technology is. We can do genome editing without any selection and achieve about a 30% targeting rate. So we can get homologous recombination at 38% efficiency. That's to put in the programmed mutation. We can get these other types of frame shift mutations at 30%, and we have unaffected cells about 30% of the time. I won't go through all the data. I'll just summarize that these isogenic cell lines that differ only at the TAS locus recapitulate all of the metabolic findings of the Barth patient-derived cells. So that really gave us confidence that we didn't just screw up the Barth patient cells or that we're looking at inter-individual variation. We're really looking at the phenotype caused by the TAS mutation. So next, we wanted to get into the cardiomyopathy. Why do these cells not contract? Or why do the patients have cardiomyopathy? Can we model that in these cells? So first, we look at assembly of sarcomeres. Sarcomeres, as you know, are the fundamental contractile unit of cardiomyocytes. Now, if you plate cardiomyocytes on an unpatterned surface, they don't really like it. And they'll look like this. They don't look like adult cells, which are nice and rectangular. They form this kind of fibroblast-looking cell. The sarcomeres, which are stained here, don't orient in a uniform direction. They're kind of parallel to the boundaries of the cell. So we collaborated with a bioengineering group, and basically they made a stamp. The stamp is of a fibronectin, which the cells love to sit on. So if you make a stamp that's a rectangle out of fibronectin, then the cells will form a rectangle. And you can see the rectangle really helps the cells assemble their sarcomeres. So these sarcomeres are oriented perpendicular to the long direction of the cell, just like in a normal myocyte. You'll also see in here, this is the Barth syndrome cell. It can't properly assemble its sarcomeres. If we put the TAS gene back, we can fully rescue the sarcomere assembly defect. And this condition, which is, you can't read, but it says glucose. So these were all done in galactose, and this is done in glucose. So this tells us that the sarcomere assembly defect is not because of an energy depletion. And the nice thing about working with engineers is they're always extremely quantitative. And so they developed an unbiased two-dimensional mathematical method to quantitate this. And I can't explain the math to you. It has something to do with Fourier transforms. I don't really get it. But we can really, in an unbiased way, show that this is disorganized. Again, we use the isogenic cell lines to show that this phenotype is specific to the TAS mutation. And again, we quantitate it and show that the isogenic lines have a sarcomere assembly defect, but the control lines don't. The TAS-modified RNA fully rescued the phenotype, And also, all of these experiments were done in glucose, so again, suggesting that this is not a phenotype due to ATP depletion. Okay, so now we can see the contractile units don't form properly, but we wanted to measure actual contractile force production. And for this, we turned to this technology known as hard-on-a-chip, which is something that my collaborator, Kip Parker, has been working on for over 10 years. And actually, he's across the street, but he was using rat myocyte, newborn myocyte models, and I met him at a meeting in Stockholm, and we decided that this would be a great use of his technology. So basically, the technology is you have a plastic sheet with known physical properties, and on the sheet, you put a patterned surface, again, of fibronectin in lines so that you make the myocytes line up. So now, since the cells are on one side of the sheet, if they contract, they'll curve the sheet, and because you know the properties of the sheet, you can then calculate the force production. So this is what the assay looks like. The red thing is just a tracker that helps you see what's happening to the sheet. It's a computer-generated tracker. Then, of course, they're very quantitative. We can measure the diastolic stress, the systolic stress. The difference is the twitch stress. So here's the control cells, and then here's the Barth syndrome cells. And you can see the Barth syndrome cardiomyocytes are really not producing much force. It's hard to see the movies actually running, but the numbers are going, so the movie is running. If we put the TAS gene back, we can fully rescue the force production. So again, this is a quantitative assay. We can show that the Barth cells have defective force production fully rescued by reintroduction of TAS. And again, the phenotype is not dependent on ATP depletion. This ATP independent phenotype is very interesting, and this data is from Richard Kelly, which shows that a patient who had severe dilated cardiomyopathy at the time of this study had normal high-energy phosphate levels, whereas other patients with other mitochondrial cardiomyopathy had abnormal high-energy phosphates. I think this is a little different from what Todd Cady showed yesterday, so it'd be interesting to see how this holds with higher patient numbers. Again, we use the isogenic cell lines to confirm that this contraction phenotype is due to the TAS mutation. So here's the isogenic control, the mutant cell line. This is with treatment with the TAS mod RNA, and again, the mutant cell line is rescued by the TAS mod RNA. And again, quantitation of this phenotype. So finally, we think we've got a well-controlled model of Barth syndrome. So now we wanted to see if we could get some new insights into treatment or pathogenesis. And these experiments started with just testing some things that had been proposed in the literature that might be useful in Barth syndrome. So we have linoleic acid, bromoenolactone, the PLA2 inhibitor, and high dose of arginine and cysteine. And I won't go through all the data, but I'll just say that linoleic acid seemed to have a little bit of effect on the maturation of cardiolipin, and it seemed to improve the ATP content. We looked at linoleic acid and different metabolic parameters, and we found that it improved the basal oxygen consumption. It improved the specific activity of the ATP synthase. It partially improved the maximal oxygen consumption. And it partially improved the sarcomere assembly. In the hard-on-chip assay, it had a beneficial effect on contraction of the myocytes. So we were thinking, what could be the mechanism of action? We originally thought of using linoleic acid because we thought it might increase cardiolipin synthesis through alternative pathways, and we saw it had a weak effect there. But it also has this unsaturated fatty acid, which makes it a ROS scavenger. So we thought, well, maybe we better test to see if its mechanism of action is through scavenging ROS. And as you know, the electron transport chain is surrounded by cardiolipin, and in the absence of cardiolipin, it likely leaks electrons, which generate reactive oxygen. So first, we confirmed that there's excessive reactive oxygen generation in this model, and in fact, the Barth cells do have a very high level of lipid peroxidation, which can be suppressed by the TAS mod RNA. It was also suppressed by linoleic acid. So we thought we better take an unrelated molecule and see if that, an unrelated ROS scavenger would have a similar effect. So we used this compound Mitotempo, which is a mitochondrially targeted ROS scavenger. And first we showed using mitosocs that the Barth cells, again, had high ROS levels that were suppressed by Mitotempo. And this was also true in the genetically engineered cells. So we looked at the effect of Mitotempo on sarcomere assembly, and we showed that it rescues sarcomere assembly in both the patient-derived cell as well as in the isogenic controls. And finally, we did the hard-on-chip assay to see if this could improve their contractile function. And we were pretty impressed by the improvement that we saw, Mitotempo treatment of the H line. It was actually equivalent to the TAS-modified RNA. So in summary, we made a iPS-derived cardiomyocyte model of Barth syndrome. We think that the combination of this iPS technology with genome editing, gene replacement, leads to a potent platform that we can use to study human cardiomyopathies. And we showed that this assembly into a tissue on this hard-on chip can recapitulate the genetic rescue of, or pharmacological rescue of these phenotypes. And we identified ROS production as a potential mechanism that links the TAS mutation to the contractile abnormalities of these patients. And we hope that maybe ROS scavenging may be a therapeutic target for this disease. So I want to thank the enormous amount of collaborators that contributed to this work. Gong is the postdoc in my lab that did all of the stem cell work. Kip Parker and Megan McCain did all of the bioengineering. The modified RNA technology came from Ken Chen and Lior Zhangyi. The Church Lab helped us with the genome editing. There are many collaborators, including Fred Voss and Chuck Murray, who are also instrumental in this study. Thank you very much.
Thank you. That's such an elegant piece of work. Should we start with you?
Excellent work as always. Just a quick question. Microphone, please.
I'll pause.
Thank you. So wonderful and insightful talk as always. And I just have one question though. Is defective sarcomere assembly a feature of Barth syndrome hearts?
That's a great question. We had one patient who underwent transplant, and we were able to look at that patient's heart muscle, and at least at the level we were able to investigate, we did not see a sarcomere assembly defect. I think, so how do you interpret that in the context of this study? I think these cells obviously are not normal cells, and I think they're in a highly stressed condition, an abnormal environment, and I think that they're manifesting a very severe defect in their sarcomere assembly, which perhaps is magnified of what you see in a patient's heart, and that those heart muscle cells are having trouble assembling sarcomere, but they're able to compensate for it.
Great piece of work. And I have two questions. The first one is this model mimicking the embryonic heart or the adult heart, do you think?
So these myocytes, again, are not making it to be adult myocytes. That's a barrier in the field right now. We think that these are similar to neonatal myocytes. So I'll give you one example that's relevant to this study. We treated these cells with oligomycin, which, as you know, will block ATP synthesis from the mitochondria, and they beat just fine, which is further evidence that the ATP production is really not the problem here, at least in this model. Because normal myocytes don't even need their mitochondria to beat in this model.
And the second question is, when you use linoleic acid to do the rescue, you rescue both the monolysyl cardiolipin ratio and the ROS.
Yes.
And when you use mitotempo to do the treatment, of course you reduce the ROS. So what happened to the monolysyl cardiolipin there?
It was also rescued. So the cardiolipin was actually more. Wait. Actually, now I'm forgetting that now. Actually, if I recall correctly, we redid many of the seahorses assays, and it did not actually correct the metabolic profile of the cells. That's right.
But the lipid profile,
I think it did not. I think it did not correct the cardiolipin profile.
Okay.
So thank you so much. This was fantastic work, and congratulations. So now there's a saying that the winners cannot be judged, but they can be asked. So I want to ask you about a couple of things. So first of all, with linoleic acid. So superoxide that you detect with mitosocs cannot directly attack linoleic acid. Their activity is miserable of superoxide towards linoleic acid. So your assumption that superoxide will oxidize linoleic acid and then prevent something is not necessarily very reasonable. So you have to come up with something entirely different. Also, linoleic acid as such, it's kind of giving these cells a piece of soap, detergent. So linoleic acid by itself cannot protect. So it will have to be esterified, integrated into something else that will then be protective. And what this something else will be, it's kind of, we can guess only. All right, so this by itself, it's not going to help. So, but,
well, I appreciate
but
that.
But obviously you see protection. That's why I said that
right.
The winners cannot be judged. So, but there may be some thinking on these subjects. So finally, with Mitotempo. So Mitotempo is a great scavenger of electrons, and I think we had a chance to talk with you briefly, so that it will grab electrons from both excessively formed electrons that leak to oxygen and produce superoxide, as well as directly from electron transport chain. So whereas the balance between the two is good to know, and it's very feasible to find it out, because Mitotempo represents a remarkably convenient tool to measure the nitroxide radicals by electron paramagnetic resonance. Okay, and so then, dependent on how reduction of this Mitotempo will be happening, you will be able to see the quenching of the EPR signal from Mitotempo. So this is done by this EPR spectrometry, but if you're interested, I have 2 of those in my laboratory, so it would be easy to establish.
So we would be interested, and I also appreciate the comments on the lipids. I would be the first to admit that we are novices on lipids, and so so it is interesting what you said about the linoleic acid, because that would suggest there's a second independent mechanism that we haven't gotten to yet.
Again, a question about lipids. You have measured for hydroxynonenal as a marker for lipid peroxidation. It is true?
Yes.
So have you been also checking for truncated phospholipids?
No. So we...
And not, in other words, which would be the phospholipid releasing the 4-hydroxynonenal? Would be interesting to check.
I see. So again, we're novices, and the reason we checked that is because we could buy a kit that could measure it. So.
Yeah, but another thing one could test is where the linoleic acid actually ends up. You know, one could analyze molecular species of cardiolipin or other phospholipids to see whether they actually incorporate linoleic acid. You know, since the mechanism, as Valerian said, is somewhat mysterious. You know, but I have another question that I think one of the most interesting aspects of your work is that the contractile elements are so disorganized, you know. And since we think of Barth syndrome as a mitochondrial disease, so do you see, what is your, if I want to provoke you to speculate, so what do you think is the mechanism of that effect?
I think the most straightforward mechanism would be to hypothesize that there's either a ROS-activated kinase or the sarcomeres themselves, which are being modified by the ROS, which would lead to a hypothesis that the ROS is being turned into peroxide that's escaping the mitochondria and affecting the sarcomeres. So that's been well described in ischemia reperfusion models, but whether that's the case here, we don't know. And we're actually testing that hypothesis by using a cytosol-directed catalase.
There's another interesting aspect that is the structural organization of sarcomeres and the spatial organization of mitochondria in well-developed, mature myocytes.
Yes.
There's a lot of organization there.
Yes.
And so in fact the mitochondria are sort of wrapped around these sarcomeres.
So in these myocytes are not so organized. So we did EM on these myocytes, and you don't see that that beautiful interdigitation of mitochondria and sarcomeres. And even in the highly organized model that we showed, they're still not organized to that extent.
Just one quick comment. Linoleic acid is the primary target of reactive oxygen species, and that's why cardiolipin and other mitochondrial problems is damaged. Therefore it's a great antioxidant if you simply add it to a cell culture. So the linoleic acid could be just simply acting as an antioxidant. It traps all the reactive oxygen species.
Well, that's what we did, but we were trying to distinguish its mechanism of action by either promoting cardiolipin synthesis or suppressing ROS. But I guess it's not a great ROS suppressor, as we've learned.